Hello, welcome back. We are moving on to the nephron loop. So we just got done covering the reabsorption and secretion of the PCT. Now we're going to take a look at what happens in the nephron loop. Very cool stuff that's happening in the nephron loop. And I might revert to be calling it the loop of Henle just because that's how I learned it. Kind of rolls off the tongue a little bit easier than nephron loop, but I'll try. Okay, so this is our overview picture. Again, I'm just showing this for the beginning of every one of our physiology lessons. So we went through this already. This was our filtration. We are using the blood pressure and the filter size, pores, and slits to filter stuff out. And then we went to our PCT. We just got done talking about that. It is here that we reabsorb mostly water, glucose, organic solutes, sodium, bicarb, and water. I already said water. And then we can secrete some hydrogen ions. So now what we're going to be doing is we're going to be taking a look at this nephron loop, this long, skinny loop that does an amazing process of what's called countercurrent multiplication. So we're going to take a look at that, and we'll keep track of what is reabsorbed and secreted along the nephron loop. Okay, so before we get into this idea of countercurrent multiplication, we have to understand some features about the different parts of the nephron loop. So it's a little bit of anatomy, but it drives the physiology. So we call the nephron loop the descending limb. So that's this side here. Descending goes down. And then we have our ascending limb here. So it's descending because that's the path that the tubular fluid takes. And then it turns that little hairpin turn and ascends, comes back up towards the renal cortex. All right, so that's where those terms come from, descending and ascending limb. Now, of the cellular characteristics, the descending limb is permeable to water, but impermeable to solutes. That's kind of like this red outline. So what that means is water can move through, which is polyaquaporins, but solutes can't. And the solutes primarily we're talking about are sodium and chloride and potassium ions. So what that means is they don't have or have very little leak channels or facilitated diffusion channels. If they're not there, these charged polar ions cannot diffuse through the plasma membrane. That's the selective permeability of that phospholipid bilayer. Water can because there's aquaporins. Ions can't because there's no channels. Okay, now if we turn the corner and come back up to the ascending limb, we see that it's kind of reverse. It's impermeable to water, which means it's not going to have any aquaporins, but it is permeable to solutes. And so we can see the sodium and chlorine, and we'll see potassium in the next slide, kind of their exchange purposes. So now we have those channels there. We can move these ions where we need them to go. So because of these different characteristics of the descending limb, permeable to water but impermeable to solutes, right next to it is the ascending limb that is permeable to solutes and impermeable to water. What this is going to do, it's going to set up a self-driving mechanism for reabsorption of salts and water. It's so cool. And pretty much passively, it's kind of amazing. So to begin this, we have to understand some of these numbers. The milliosmoles per liter. This is just a unit of concentration. So it's osmolarity, it's kind of osmotic, the concentration of things that are dissolved in the watery environment. It's just a unit of concentration if you want to think of it that way. So by the end of the PCT, the tubular fluid has an osmotic concentration of about 300. Okay? As it goes down in the tube, we can see it's getting more and more concentrated, 300 to 600 to 900, all the way down to 1200 milliosmoles. And then on the way back up, we can see that it's getting less and less, and it actually leaves the loop of Henle at 100 milliosmoles. So from going in to exiting the loop of Henle, we've lost concentration. So which means we've reabsorbed, we've reclaimed some of those solutes, which is part of the job of the nephron loop. Okay, so that's inside of the tube. Now, if we take a look at what's going on outside the tube, we see some very similar numbers. So this is what's happening in the medulla. So this is the fluid and the tissue that's outside of the nephrons. We can see 300, 600, 900, and 1200. And you might be saying, hey, those numbers look almost exactly the same. And you are correct. They're very similar to the numbers that are inside of the nephron loop. And the reason for that is that whole idea of diffusion and osmosis and equilibrium. We cannot make urine that is any more concentrated than the concentration out here in the renal medulla, this extracellular space, because you can't move water into an area that already has a lot of water. So that if to do that, to make more concentrated urine, we don't pump water. We don't have that ability to do that. So the concentration of our urine is going to be based on the concentration of this medullary space, the concentration of the surrounding fluid, if you will. So let's take a look at what happens. This is our countercurrent multiplication. So as the tubular fluid comes in to the thin descending limb, right, it's permeable to water but impermeable to solutes. So if we take a look at the osmotic differences, as we go down, the water is going to be drawn out of the tube because it's permeable to water because it's trying to go to that more hypertonic environment outside of it. As it does that, the solutes cannot move. So the solutes get left behind, but the water gets reabsorbed. So it gets moved out of the tube. And so if we lose more and more water, what's going to happen to the concentration of our tubular fluid? Well, it's going to get really concentrated as the environment outside of it. So it's going to reach an equilibrium. As the water leaves, the inside of the tube can only reach about 1200 milliosmoles. That's pretty concentrated. So then it turns the corner. No more water can be reclaimed, but now we have these really high concentrations of ions that can drive their movement out. Remember, water is impermeable now. And so here we are going to be pushing the sodium and chloride out of the tube. And if we can see, our concentrations are getting less and less, until we leave the nephron loop at 100 milliosmoles. Now, all the sodium and chloride that's getting pumped out of the nephron loop helps to set up this gradient in the medulla, which, if we go back to the beginning, drives the water reabsorption on the descending limb, which drives the concentration here, which drives the reabsorption here, which drives the water reabsorption. So you kind of get into this loop of water reabsorption drives solute reabsorption, which drives water reabsorption. And that's this whole idea of countercurrent multiplication. Countercurrent because they're going in opposite directions. Then descending limb goes down, ascending limb goes, and they're right next to each other. Multiplication is because the action of one of the limbs drives the action of the other, which helps to support driving of the first one. So it's kind of this positive feedback almost mechanism, but not technically. So that is the understanding of countercurrent multiplication. Now, if we take a look at this picture over on the right-hand side, we can kind of see the same thing. We're reabsorbing water. We're going to be reabsorbing solutes. But if we take a look, now we're bringing in the collecting ducts. So the DCT and the collecting ducts here. Now, if we leave the nephron loop like we did up here, we're going into the DCT, it's at 100 milliosmoles. But that's really dilute. We don't want to pee out urine that's 100 milliosmoles. That's a waste of water. And if we take a look, what we actually do release urine is at that equilibrium with the medullary space of 1200 milliosmoles. So how do we go from the end of the nephron loop at 100 to urine that's at 1200? Well, we have some other characteristics of the DCT and the collecting duct and the papillary duct that allow for further water reabsorption while leaving the solutes in the urine. So here, the very ends of the collecting duct, this what's called the papillary duct, is permeable to urea. Now, urea is one of the nitrogenous wastes that we like to get rid of. So you might be saying, well, why are we wanting to be permeable to urea? Why do we want to let some of that urea come back out into the deep part of the medulla? Well, what that allows our kidneys to do is to keep that deep part of the medulla really concentrated. So we want to keep this nice and hypertonic. And the urea helps to maintain that hypertonic environment, which will help to drive further reabsorption of water at the collecting duct and the papillary duct. So we are reabsorbing water back into our peritubular capillaries, and then we get our final concentration of urine, which is around 1200 milliosmoles. So in a way, the urea helps us to reabsorb water. So it's okay that we allow a little bit of it to leak out because it helps to maintain that concentration in the renal medulla, which helps drive the water reabsorption, which helps drive the sodium reabsorption there in the ascending limb. It's pretty cool. So that's countercurrent multiplication, the role of the descending and ascending limb based on their differing characteristics of the membranes, right? Permeable to water, impermeable to solutes, and reverse. And then the role of urea to help maintain that concentration outside of the collecting duct to help reabsorb the last little bit of water before the urine goes into the calyces. All right. All right, so this last slide is showing just the details of what's happening in the ascending limb, the part of the solute reabsorption. So we just learned that the descending limb is water reabsorption. We can see here. Okay, so water is being reabsorbed, concentrating the solute down here. And then as we make our way back up the ascending limb, here's going to be our channels. So we can see our green little circles are co-transport. So if we take a look, we are reabsorbing chlorine ions, potassium ions, and sodium. And so this particular co-transport, 3 ions are being able to be co-transported from the tubular fluid into the cells. They diffuse, and then we get some co-transport with chlorine and potassium and some active transport with a sodium potassium pump. Okay, so if we want to keep track of what is reabsorbed, kind of like what we did on the PCT, we have chlorine, we have sodium, and we have potassium. Okay, so that's what's reabsorbed. If we want to keep track of what's secreted, right, we have potassium because it's right here. So potassium diffuses and has a leak channel to go back into the tubular fluid. So we can basically, in essence, cancel out potassium. It's just along for the ride. So potassium helps to come in with the co-transport of sodium and chloride. It helps with the co-transport of chlorine, but then it turns right back around and plays this role of the active transport of reabsorbing, kind of the pumping of the sodium and the pumping out of the potassium. So potassium just kind of goes in this loop-de-loop, playing right back around here. So really, the main thing that we are doing in the nephron loop is reabsorbing chlorine ions and reabsorbing sodium, right? So these are salts over here. But what do the salts help us reabsorb? Okay, one thing I need to add to my list over here is water, right? So water and salt. That's the major job of the nephron loops. Reabsorb sodium, reabsorb chlorine, and that water follows that in the descending limb. Salts reabsorbed in the ascending limb, which drives the reabsorption of water in the descending limb, which drives the reabsorption of salts in the ascending limb, which drives the reabsorption. You get the picture, right? So it's just this continual multiplication of water and sodium reabsorption. It's awesome. Okay, let's move on to the distal convoluted tubule in our next video. Bye.